I would like to introduce myself. I am the Chief Business Officer of Sino Biological, and it is my pleasure to officially welcome everyone online now to the next installment in our series on COVID-19 cutting-edge immunodiagnostics. So welcome everyone. Good afternoon, good morning, or good evening to you, wherever you all may be. A few housekeeping items. I want to mention that everyone here will be muted throughout this seminar, but you can ask questions in the chat box, and I highly recommend you type in your questions into the chat box. And at the end of the seminar, I will select questions from the chat box and ask our speaker today to answer those questions. In addition to that, I want to mention that this webinar is being recorded, and everyone who registered and is attending today will get a link to the recorded webinar. So without further ado, it is my pleasure and honor to introduce to you our speaker for today. Our speaker's name is Dr. Xiaotian Tan, and he is a research fellow at the University of Michigan in the Department of Biomedical Engineering. Dr. Tan studies under the guidance and direction of Professor Zhudong Fan. He received his doctorate very recently from the University of Michigan in February of this year. And Dr. Tan has extensive expertise in chemical engineering and molecular immunology, and he has been working specifically on the development of a highly selective optical biomolecular sensor system for literally the last 5 years. So Dr. Tan, the title of his talk is Rapid and Quantitative Detection of SARS-CoV-2 Specific IgG for Convalescent Plasma Therapy. So Dr. Tan, it's a pleasure to have you today. We appreciate your time, and now I will officially turn the screen over to you.
Thank you, Rob. I'll share my screen at this moment. Hello, everyone. Please let me know if you can see my screen now.
Yes.
Okay. So thank you very much for inviting me for giving this talk. The title of this talk is, I changed it a little bit, is Rapid and Quantitative Detection of SARS-CoV-2 Specific IgG for Convalescent Serum Evaluation. So I'm currently a postdoc working under the guidance of my advisor, Professor Xudong Fan. And I would like to read a few more disclosure statements on behalf of him. So the system we are using is developed in collaboration with University of Michigan and the startup company Microfluidic Bioassay. And Dr. Xudong Fan is a co-founder of this company, in which the device and the cartridge presented in this presentation are being commercialized by this Microfluidic Bioassay company. And this project is done with a collaboration from Hackensack Meridian Health, Microfluidic Bioassay, and Sino Biological. Here is the start of my presentation. So I'll first introduce SARS-CoV-2. I guess everyone knows about that already. And I'll also introduce convalescent serum therapy for critical COVID-19 patients. And I'll introduce my results or system and or quantification of SARS-CoV-2 specific IgG for serum evaluation results. I also give a brief summary and some future perspectives about this technology. So here is the introductory part. I guess, as everyone knows already, COVID-19 is caused by the RNA coronavirus, SARS-CoV-2. The structure of this virus is shown here. So basically, this virus uses a protein called spike protein to bind with ACE2 receptor on human epithelial cells. This is an invasion mechanism of this virus. So in infection conditions, the spike protein can bind with it and then goes into the cell in the human. So for COVID-19, hold on. So for COVID-19 disease, the hospitalization rate and the death rate are both proportional to age. So probably for young people like us, we are not that dangerous because the fatality rate is typically around less than 0.5%. However, when we get older, especially for the elder people with an age elder than 70 or 80 years old, their fatality rate and their hospitalization rate are very high. So, which means the risk of like getting killed by this disease is like several folds higher than the young people. So we definitely need some antivirus medication. So within the year of 2020, there are like a bunch of researchers and doctors are trying to evaluate the performance of several antivirus drugs. For example, remdesivir and hydroxychloroquine. They're pretty famous on the news, but unfortunately, both of them are having only limited efficacy for critical cases. They are definitely they can definitely give a help on the recovery of the patient, but they are still not specific medications for this disease. So we may need an alternative approach, which is the convalescent serum plasma therapy. So in this case, in this therapy, the antibodies in the convalescent plasma or convalescent serum will bind and cover the virus and stop it from invading the cells in the human. So I'll also give a brief introduction about what is convalescent serum. So during an COVID-19 infection, if the virus infects a healthy human, this person can develop and secrete antibodies to neutralize this virus, as I introduced in the previous slides, especially to bind and block the receptor binding domain of the S protein. And in the convalescent serum, SARS-CoV-2 specific IgG, especially S1 or S1 RBD specific IgG, is a major effective component. So it will last several months after the recovery. So we can use IgG as a therapeutic agent for treating critical COVID-19 patients. So the most traditional way of detecting IgG in the serum is called colloidal gold test strips. It definitely have a lot of advantages. For example, it's fast, cheap, and easy to use. Basically, just add on one drop of blood on it, and you can get results. And you can get the result within 5 to 10 minutes after you get this one drop of blood on it. However, there are also several major disadvantages of this technology, which is this device is qualitative, can only provide qualitative, and it has very limited sensitivity. So in the hospitals, the doctors prefer to use ELISA to generate the IgG measurement with a higher sensitivity, a higher repeatability, and to get a quantitative result. However, the traditional ELISA is performed in this type of reactor called 96-well plate or microtiter plate. Although it has a micro in its name, its surface to volume ratio is pretty bad. And it also requires an expensive plate reader for taking measurements. And there's other limitations, such as it requires larger sample consumptions, and it takes 3 to 6 hours to generate a result. So all these limitations make the turnaround time of the serological analysis pretty long. For the convalescent serum evaluation specifically, this is a paper recently published. The scientists found that neutralization efficacy is related to the abundance of SARS-CoV-2 S1-specific antibody. However, to measure the abundance of the SARS-CoV-2 specific antibody, multiple dilution factors is needed to generate decent titration curves. And another limitation is this type of measurement generally doesn't have a calibrator. So the quantification is only performed as using the optical density as a readout. So this is not a really quantification. So, which means a calibrator is needed. So here comes our solution. 2.5 years ago, we developed this microfluidic chemiluminescent ELISA system. So basically, we use this capillary sensor array as an ELISA reactor in replacement as a 96-well plate. In this reactor, the surface to volume ratio is about 10 times higher than the traditional plate-based ELISA. And since we have a microfluidic structure, we only need 8 microliters as a sample volume. So, and we also integrate this into an automatic controlling system. So it has a robotic arm, a liquid pump, and the CMOS camera. The reason that we choose to use chemiluminescent way for taking measurement is we want to get rid of all the unnecessary parts. If you decided to use absorbance measurement or fluorescent measurement, we need external excitation light. But for chemiluminescent, this is not required. The only measurement device we have is a CMOS camera. Oh, by the way, this system was developed in collaboration with the startup company, Optofluidic Bioassay. So for taking measurement with this system, we use a luminol-based chemiluminescent substrate to generate the measurement signal. So, and since it has a blue color, so when we take measurement, we have an image, and we take out the blue channel, and we quantify the light intensity with a software called ImageJ. So at the beginning of this, our system, first few years we have this system, we never tried to use this to clinical applications. We were basically measuring the biomarkers in small animals. For example, this one, the mouse follicle stimulation hormone. So we quantified the mouse follicle stimulation hormone from the tail vein blood, a tiny drop of tail vein blood, with the system in only 30 minutes. So it was the traditional way of quantifying this hormone is using radioactive immunoassay, which takes several days to complete. But here we can get it in 30 minutes. This is due to we have a much higher surface to volume ratio, so which means we have a higher reaction rate. And as you can see here, we can generate a much wider calibration curve, the linear dynamic range, comparing to radioimmunoassay and traditional ELISA assay for the specific markers. And we also tried to repeat the calibration experiment with multiple trials. And turns out our inter-assay variance is very small. This is due to the fully automation of our system. And the error bars are also pretty small. And with this system, and now we can use it to perform COVID-19 related applications. In this specific task, we have 3 aims. First is finding a good calibrator antibody. And second is to quantify the anti-S1 IgG in the convalescent serum. Third one is to detect the viral antigen, only as a demonstration of concept. So let's come back to this slide. Why we need to find out a good calibrator antibody? First, as I mentioned, without the calibrator antibody, we can only quantify the abundance of SARS-CoV-2 specific antibody with optical density, which means it's very hard to convert it back to an actual concentration of the antibody. And in addition, ELISA assay typically will have some inter-assay variances. So if you perform assays on 2 different days, you might get different results. So with a calibrated antibody, you can always transform your measurement result back to calibrator, the effective concentration of the calibrator antibody. So this is the reason we need to select a calibrator antibody first. So in this assay, a good calibration antibody, the requirement of a good calibration antibody is need to have a high affinity toward SARS-CoV-2 S1 protein. Second, it's need to have a large dynamic range towards this S1 protein. Third, it's need to have a relatively high specificity towards this SARS-CoV-2 S1. And in order to eliminate all sorts of errors, we first performed this assay with a single-step ELISA, which means we directly conjugated HRP, the reporter enzyme, onto the potential calibration antibodies. And I also need to mention that in this application, we didn't directly mobilize recombinant S1 protein on our capillary. Instead, we use an anti-polyhistidine antibody as a capture antibody for this recombinant S1 protein. This is because we want to make the receptor binding domain available for all the calibrator antibodies. And we also performed a side-by-side comparison with SARS-CoV-2 and SARS coronavirus. And we chose 4 calibration antibodies, potential calibration antibodies to test here. CR3022 is a pretty famous antibody now. It was an antibody that was isolated from SARS patient. And in February this year, it was reported to have cross-reactivity with SARS-CoV-2. And the other 3, D001, D003, and D006, are chimeric antibodies provided by Sino biological. They were also reported to have binding capability with SARS-CoV-2 S1 protein. And as you can see here, for SARS-CoV-2, these 4 looks pretty different. CR3022 look like it has the lowest affinity, and D006 has the highest affinity towards SARS-CoV-2. For SARS, it's a different story. All these 4 antibodies looks pretty similar, and this is because all of them were actually raised against SARS coronavirus. And in this case, D006 was the one that has the highest affinity. In fact, another reason that we decided to use this method for performing affinity evaluation is, although there are several popular ways for doing antibody evaluation, for example, SPR, biolayer interferometry, BLI, they are not really representative for the ELISA settings. In this case, we are directly evaluating their affinity in ELISA settings. However, the single-step ELISA can generate some measurement results, but it's not in a realistic settings. Remember that we directly immobilized, conjugated HRP molecules on the calibration antibodies. So we decided to test them in more realistic settings, like this. So we still immobilize the S1 proteins through this anti-His antibody immobilization, and we use some sort of sandwich ELISA structure to detect the concentration of this calibrator antibody. And here are the entire dynamic range of these 4 antibodies. Apparently, CR3022 has the shortest, the narrowest linear dynamic range. It's only between 5 to 300 nanogram per mil. The other 3 antibodies are having very similar performances. their linear dynamic range are around 2 to 1,200 nanogram per mil, which is pretty wide already, close to 2 orders of magnitude. 3 orders of magnitude. So, as I mentioned, since we are using a microfluidic system, we can complete the entire assay within 15 minutes, which is around 10 times or even more than 10 times faster than a traditional plate-based ELISA. And in this case, we are getting very sensitive and quantitative results, which is much better than the colloidal gold test strips. So from the results shown here, we can clearly see that CR3022 is not a very good collaborator, not a very good calibrator. D006 has the widest linear dynamic range to SARS-CoV-2 S1, and it also have a high affinity to SARS-CoV-2. So we decided to use D006 as a calibration antibody. And we also performed a side-by-side evaluation for SARS-CoV-2 and SARS. This also verifies that D006 was the only one that has a higher affinity toward SARS-CoV-2 S1 comparing to SARS coronavirus. So, as I mentioned, due to all this information, we decided to select D006 as our calibration antibody. We also tried to improve the detection sensitivity of our assay by replacing the S1 with the S1 RBD, receptor binding domain, as a capture antigen. But we, as I showed here, is only generate a more sensitive result, which means the slope is higher, but it cannot generate measurement with higher, a better limit of detection. And so what makes things even worse is make the dynamic range of this assay shorter. So we still decided to use S1 as a capture antigen to generate as wide as possible dynamic range. So after we found out this good calibration antibody, we initiated a collaboration with Professor David Perlin's group at Center of Discovery Innovation at Hackensack Meridian Health. So they were doing clinical trials on using convalescent serum or plasma to saving lives in critical patients. So there are procedures like this. So they identify some recovered COVID-19 patients. They collect some blood samples, and they send it to central lab and perform ELISA analysis. And from the ELISA analysis, they identify the qualified donor for the study, and then they collect the blood from the qualified donors. As you can see here, these will result in a very long turnaround time, probably 2 to 3 days at least. So we wonder if our technology can give an help on this. So this is myself, and this is Dr. Mila Krell. So we ship our microfluidic ELISA system to their hospital in New Jersey and use it to test several patient samples, including 16 samples from recovered COVID patients and 3 samples from healthy donors. So this was happened in May, late May. So for assay validation purposes, we performed a side-by-side test with traditional plate-based ELISA. Here is a plate-based ELISA result. So all the measurements were performed with a plate-based ELISA. The assay time was around 3 hours in total. And so the cutoff value was selected to be 0.2 as the optical density. So at 100 times dilution, if the measurement result is below 0.2, the samples were classified as negative samples. However, 3 samples from COVID-19 patients were identified as negative samples with this plate-based ELISA. Let's see what will happen with our system. here is a serial dilution curves with our own microfluidic chemiluminescent ELISA system. And we performed multiple dilution factors, in fact, 6 dilution factors, starting from 40 times. And we have 5 times serial dilution all the way down to around 100,000 false dilution. And from this result, it's very clear that if you want to identify the best donor for its convalescent serum therapy, we need to dilute the serum for 200 times. Here, you can easily distinguish the strongly positive samples from the medium positive samples from the negative samples. And so we decided to use this 200 folds as a dilution factor in our future assays. By the way, NC1 and NC2 are 2 negative controls. They were both purchased from Sigma. They were serum from healthy donors. And here are the results we generate from a single dilution factor, which is 200 times dilution factor. In this case, NS1 and NS2 with our system were detectable, which means they might be false positive if we compare it back to traditional plate-based ELISA. But still, we can see pretty clear difference between the 2 groups. There's definitely a statistical meaning between difference between this positive group and the negative group. As you can see here, the signal intensity varies a lot for all the positive cases. But for the negative cases, all of them are very low. And now we decided to convert this back to the effective calibrator antibody concentration. So we use this red calibration curve to convert the signal intensity shown here into effective D006 concentration. And here are the results after the conversion. Very interestingly, if you look at the result here, this is pretty clear to see that just look at the positive group. The weakest serum is like almost 100 times or more than 100 times weaker than the strongest serum, which means even from the positive recovered COVID-19 patients, their therapeutic potential for doing convalescent serum therapy is really different a lot, for more than 2 orders of magnitude. And if we want to make a selection of which donor we can use to perform this convalescent serum therapy, we need to find out a cutoff value for this. So we did some literature research, and here are several neutralization antibodies that were identified in a group from China. And most of them, the IC50 value for the monoclonal neutralization antibody were around 1 microgram per ml. So we give it some room. We choose 2 microgram per ml of antibody in the recipient serum as a target concentration. But in a convalescent serum therapy, the donor serum is typically diluted by 25 to 50 times after transplanted into the recipient's blood. This is because when you can only get 100 to 200 ml of serum out of the donor, and you need to dilute it to the total blood volume of the recipient, which is around 5,000 ml. So after the dilution, we need to reach this target concentration, which means the effective IgG concentration in the donor serum should be at least 100 microgram per ml. So we convert this back to the concentration after 200 times dilution. So we decided to use 500 nanogram per ml as a cutoff value. And here is the result after the cutoff. Although they all have some antibodies, only 4 of them can be identified as a qualified donor for convalescent serum therapy, which is a very small portion of this of the positive COVID-19 patients can be selected as a qualified donor for the serum therapy. So as I mentioned, our assay can generate results within 15 minutes, which means if we can deploy one of the system to a blood collection trial, we can probably do an on-site evaluation of their serum using a fingertip blood to evaluate which recovered patient is the best donor candidate for the convalescent plasma therapy. And then if we identify some like strong donors or super donors, we can do like evaluation, for example, neutralization potential evaluation of their serum after we identify them as a potential donor. So we do not need to do like neutralization evaluation for all donors. This will definitely speed up the turnaround time of finding qualified donors in the convalescent serum therapy, and definitely will give some help to the throughput of the convalescent serum therapy. So after this, I also want to introduce briefly regarding the detection of some COVID antigens. Before that, I'll also introduce our own enhanced sandwich ELISA assay for detection of antigens. In this case, I'm presenting this IL-6 detection data with poly-HRP amplification. And with this, you can see we can reach the detection limit down to 0.3 picogram per mil level, which is a pretty remarkable detection limit, considering we are only using analog ELISA instead of a digital ELISA. So you may wonder why we have such a high detection limit and a wide dynamic range. This is because we have a technology called multiple exposures. So for high concentration samples, we do exposure at a very short exposure time. But for low abundant samples, we can make the exposure time of the chemiluminescent signal much longer to get very weak signal out of the camera. So with this poly-HRP amplification and the multiple exposure technology, we give a try on the detection of SARS-CoV-2 antigens. So all the kits were provided by Sino Biologicals, and it's very obvious that we can get down to 4 picogram per mil of S protein and down to around 60 picogram per mil of N protein as a detection of SARS-CoV-2 antigens. This is only a demonstration of concept study. We haven't got the chance to test it in real clinical samples. So here is a brief summary and some future perspective of our assays. So first, we demonstrated the sensitive and rapid detection of viral antibodies, especially IgG. The assay can be completed in 15 minutes with only 8 microliter of sample. So we identified a humanized chimeric antibody, D006, which can serve as a potential calibration standard. And we also applied this technology to the selection of best donor of the convalescent serum therapy. And as I mentioned, only 4 out of the 16 recovered COVID-19 patients can be identified as the potential donors. We also briefly demonstrated the potential in detection of viral antigens, especially S and N proteins for COVID-19 diagnosis. The entire assay can be completed in 40 minutes with picogram per mil sensitivity, which is much better than traditional ELISA. So in the future, we are also planning to do a few more things. First is evaluating more potential calibration antibodies. D006 is definitely a very good antibody, but it's still not a perfect antibody. The optimal antibody for the convalescent plasma therapy is an antibody that's isolated from recovered COVID-19 patients. So if we can identify a good calibrator antibody in the future, we can use that as a calibrator antibody. It's very easy replacement. So, and another thing is we may be able to do the viral neutralization assay with our system as well. We are also planning to detect viral particles directly with our ELISA assay. So with this, I would like to finish up my presentation. I would like to give special thanks to my professor, Dr. Xudong Fan, who is not only an advisor, but also a lifelong mentor to me. I learned a lot from him. And I also like to thank my collaborators from HMH, Professor David Berling, and Dr. Mila Krell, Dr. Enrico Dogolf, and Mr. Stephen Park. And I would also like to thank my collaborator in optofluidic bioassay, Dr. Malcolm Tiang Wu, for developing this microfluidic chemiluminescent ELISA system. I would also like to thank Sino Biological for providing the reagents and providing some discussion with me. Thank you very much for joining me.
Thank you, Dr. Tan. That was a wonderful seminar. We appreciate it. It was very data heavy. We appreciate you taking the time to present your exciting platform, your technology, and your results as well. I want to remind the audience that you can type your questions into the chat box here, and I will do my best to get them answered by Dr. Tan. I'll go ahead and jump right into the question and answer session if I can, and I'll start with a question from a researcher by the name of Arun Agarwal. And Arun is asking just a technical question regarding the difference between D006 and the CR, 3022 antibodies. Could you just clarify why D006 was so much better?
Hold on a second.
Sure.
I would say probably the difference is caused by the binding epitopes and the affinity toward the SARS-CoV-2. All of them were actually raised against SARS in fact, but some of them, they are binding to the common epitopes between SARS and the SARS-CoV-2. Some of them, the epitope changed in the COVID. So this is the reason that CR3022 has like a significant drop in affinity. But the others doesn't have a very significant difference between these two.
Great, thank you. And we're jumping right into the business aspect, I guess, of the technology. And a researcher by the name of Ashraf El Minaway is asking about the systems themselves from optofluidic bioassays. And are the systems available for purchase? If so, what's the list price? If you could provide that, just tell us a little bit about the system's availability.
I am not involved in the business, so I would suggest to contact Dr. Malcolm Tiang Wu. You can find his contact information on the company's website.
Okay. Can you mention the company's website just so all of our
just search optofluidic bioassay in Google.
Optofluidic bioassays.
Yes.
Okay. Okay. We have to plug the company since they're your collaborator, right? So let me ask a question. I mean, obviously you focused on coronaviruses, not just COVID-19, but another one. Have you studied any other types of diseases using the system?
Not really. We never applied our system into a clinical study before, in prior to this COVID-19 pandemic. But we did some measurement with animal models of cancer, for example, bladder cancer. We had a recent publication regarding using a urine marker concentration to model the progression of bladder cancer.
And here's a question, it's not really related to the optical technology, but it's more about the coronavirus itself. I mean, do you see any advantage to focusing only on the spike protein and not necessarily on the N protein or maybe other proteins? Is there a value in detecting like anti-N protein antibodies as well as anti-spike protein?
It's depending on the purpose of this application. For hold on, let me get back to the first slide. Yeah, the M protein is basically on the inside of this virus. So if you want to detect it for therapeutic purposes, I would say still anti-S protein and IgG is more important. But if you want to do it for diagnostic purposes, then probably anti-N antibody is also very important.
And here's another technical question from Shakil Farouki. It's really about the correlation between the very sensitive picogram amounts that you're detecting of the SARS protein with viral load. Can you do a direct correlation between the amounts that you detect and the actual viral load or the number of viral particles present?
Unfortunately, we never did clinical evaluation with the detection of viral antigens. We were focusing on detection of viral antibodies at first. We only have some demonstration of concept results for the viral antigen detection.
Okay, okay. Let me just ask a question. I'm sure you've already answered this, but I'll ask it myself. You use a CMOS camera to detect the chemiluminescence, is that correct?
Yes.
Okay, and can you just tell us the advantages of that over like a typical ELISA plate reader, which is also pretty sensitive?
Typical plate reader, if you are using a absorbance measurement, some of the high concentration results will saturate very easily. So the dynamic range is shorter than chemiluminescent plate. So in our case, because the light intensity is proportional to the concentration. So if you have a high concentration sample, we can just reduce the exposure time. So which means we can have extended dynamic range on the higher end.
Okay, great. And I'm assuming it's really the microfluidic aspect of your technology, which obviously allows for the very low, like 8 microliter sample volume, then, right?
Yes, yes, indeed.
Great. Well, that sounds good, Dr. Tan. I think, oh, here's a question. We still have some time left, so let me ask this. I'll go ahead and just ask it directly from the chat box. It's from Jonas Boating, and it's how do your concentration ranges correlate with titers? Oh, I guess you just kind of already answered that. But it says, for example, the FDA recommends a 1 to 160 titer for convalescent donors. Where does this titer fall within your concentration range? You kind of answered that already. Do you want to elaborate on that?
So in fact, our titration curve is not really comparable with traditional plate-based ELISA results. Hold on. The reason is this is a plate-based ELISA result, and this is our zero dilution result. The reason that we have a different result is we are using a very short incubation time. So, and within our incubation time, we can, the strong antibodies dominate the binding process. But the weaker antibodies, they do not have a chance to bind with the antigen yet. So I would say, if you want to correlate our results with the titer from plate-based ELISA, we may need to do some additional experiment.
Great. Another good technical question here, just really quickly. It's from Richard Kababwe, and it is, how do you ensure that the inherent noise in the exposure radiation does not give a false result?
We have a very good blocking technology, so and we do not observe a very strong background noise in most of the cases.
Yeah. And since it requires a very small sample volume, I mean, would this work for saliva or other biological samples?
Definitely, we give a try with saliva for measuring other markers other than the viral-related markers.
That's great.
No problem at all.
That's great.
We can even measure the markers from the mouse urine and mouse tail vein blood, because they're all having very limited volume.
Great. One more question. I believe it's relevant. It's from Nagora Babeva, and it is, does protein composition matter in this study? I'm not sure what that's meant, but maybe you can elaborate.
I do not quite get that.
Yeah, that's all right. Well, with that, we will finish this in the series on COVID-19 Immunodiagnostics webinar. And Dr. Tyne, I want to sincerely thank you for an excellent seminar. We really appreciate it. It's a wonderful technology that you've developed in collaboration with optofluidic bioassays. You're a great partner and collaborator, as is the company. And we appreciate.
Thank you very much for inviting us to give a talk here.
Thank you, and we appreciate your time today, and want to thank all of the attendees. Remember that you'll get a link to this recorded webinar as well. And then finally, I want to thank my good friend and colleague, Dr. Jen Liu, who coordinated and executed this as our marketing manager today. Thank you very much, Jen. And with that, I'll close the meeting today, and I'll say goodbye, and everyone.
Thank you, Rob, and thank you everyone for attending the talk.
And evenings. Thank you, Dr. Tan. Thank you again. Bye.